Good morning. It's March 3rd, and this is your daily brief in science. Here's everything you need to know. A new approach in the fight against type 1 diabetes targets the insulin makers themselves rather than the immune system at large. University of Chicago researchers report delivering PD-L1 mRNA directly to beta cells using an mRNA-based nanoparticle therapy, with the aim of shielding these cells from autoimmune attack and inflammation. In early animal tests, the therapy reached beta cells and delayed diabetes progression in mice, including models with transplanted human beta cells. The work is a proof of concept published in Cell Reports Medicine and does not include human data yet. Lead author Jacob Enriquez and co-author Raghu G. Murmura note the novelty of cell-type-specific delivery and the potential for targeted immune protection of beta cells, offering a contrast to strategies that broadly modulate the immune system. The study was funded by Breakthrough T1D and the NIH and represents a first step toward precise, beta cell-focused prevention or delay of type 1 diabetes. Questions remain about long-term safety and how long protection might last, as no human trials have occurred to date. A surge in measles cases has put vaccination front and center, with acting CDC Director and NIH Director Jay Bhattacharya urging Americans to get the shot as the best protection for individuals and communities. Bhattacharya, stepping in as acting CDC Director in February, emphasizes vaccines amid ongoing outbreaks and rising case counts across the United States. Healthcare providers are urged to help families make informed vaccination decisions, even as debates over informed consent and shared decision-making linger in policy discussions. The messaging reflects a balancing act between public health leadership and shifting vaccine policy and outbreak response. A Reuters/Ipsos poll finds a bipartisan majority believe vaccines are safe and that children should be vaccinated to attend school. South Carolina has sought non-CDC reinforcements to manage the outbreak, temporarily expanding its epidemiology workforce and requesting analytical support from infectious disease scientists, with personnel support from the CDC Foundation. PAHO plans an April meeting with the U.S. and Mexico to review measles statuses and share data, as the United States risks losing its measles eliminated status since 2000, with January 20th cited as the date by which the status wasn't met. Vaccination is framed as essential for individual and community protection, aligning with core CDC priorities to protect the American people with integrity and accountability. Bhattacharya stated in a video posted on X that the CDC is increasing resources and coordinating with states to educate and outreach to contain measles. Claudins are moving from background players in tight junctions to actionable targets in cancer therapy, with their roles in barrier function and signaling across multiple tumors driving both progression and new treatment approaches. The landscape now includes CLDN 1.2, CLDN1, and CLDN6 advancing toward clinical development, as researchers shift toward using Claudins for diagnostics and targeted therapies. Overexpression of Claudins in solid tumors is linked to growth, EMT, cancer stem cell traits, and drug resistance, making them central to how cancers progress and respond to treatment. Understanding resistance through mechanisms like Claudin isoform switching and junctional remodeling is essential for designing combination strategies that sustain efficacy. Key mechanistic insights show tight junction components interacting with PDZ domain proteins to regulate polarity and influence signaling pathways and EMT. Claudins also affect tumor immunity, offering potential synergy with immune checkpoint inhibitors to boost anti-tumor responses. Integrated diagnostics, including immunohistochemistry, RNA in situ hybridization, and sequencing, are needed to accurately profile Claudin expression for precision medicine. The dysregulation of Claudins disrupts signaling pathways such as AKT and MAPK and shapes the tumor microenvironment, influencing immune infiltration and extracellular matrix dynamics. Heterogeneity in Claudin expression across cancers and within tumors poses a challenge, underscoring the need for isoform-specific biomarkers and robust companion diagnostics. Advances include structure-function studies, Claudin-specific antibodies and ADCs, and preclinical reports of Claudin-targeted imaging and modulation of fibrosis and inflammation. Therapies exploit Claudin extracellular domains for targeted delivery, while exploring intracellular targeting to modulate downstream signaling and improve durability. Targeting intracellular Claudin pathways and protein interactions can complement extracellular antibody therapies, addressing extracellular heterogeneity with an additional modality. A Stanford-led study reframes math difficulties in children as a challenge in learning from mistakes and adjusting thinking after errors, not just raw number processing. Researchers tracked second and third graders solving larger number problems, measuring response times and brain activity with MRI to link behavior to neural signals. When problems were shown with dot arrays instead of Arabic numerals, the groups behaved more similarly and showed similar brain patterns, suggesting representation matters for early math learning. The study identifies lower activity in the middle frontal gyrus and anterior cingulate cortex among children with math difficulties, brain regions tied to number processing, focus, error detection, and performance monitoring. A key finding is that differences in post-error adjustment distinguish typical from struggling math learners, pointing to broader cognitive control issues beyond numerical skills. The work implies interventions should cultivate metacognitive strategies and alternative problem-solving approaches, not just strengthen basic number sense. The findings align with reports in the Journal of Neuroscience and Stanford News on observed brain patterns and educational implications. In parallel, a National Center for Education Statistics survey of about 30,000 ninth graders found that fair and effective teaching correlates with stronger math identity and future engagement in mathematics. The study also used a model to track performance shifts over trials and number comparison tasks using digits and dot arrays, highlighting how consistency and strategy adjustments evolve over time. Experts emphasize that fair, effective teaching can shape students' attitudes toward math and influence course and career choices, underscoring the role of classroom practice in learning outcomes. A warning from the University of Queensland says a fungus called Nanisiopsis barbati is threatening Australia's reptiles, causing skin lesions, lethargy, starvation, and death as it spreads through the environment. The study says the risk could be as serious as other wildlife fungal diseases and could affect native species across the country. Researchers emphasize environmental transmission and urge nationwide monitoring, proactive management, and development of control and prevention strategies to curb outbreaks. Since its first detection in 2013, infection rates in the studied population have risen by about 35% over 10 years, marking a growing threat. Public involvement is encouraged, with residents asked to report sick water dragons and researchers welcoming photos or videos, while authorities are asked to provide support to protect reptile populations. NB was first found in wild water dragons in Roma Street, Parklands, in 2013, 4 years after its initial discovery on a pet lizard, and has since affected roughly half of the park's about 400 dragons. There is no current cure, and climate change and habitat loss may worsen the disease's impact, prompting calls for cross-government collaboration to understand and mitigate the threat. Researchers remain uncertain whether NB is native to Australia or introduced, with national distribution unclear. Soil samples in Brisbane have tested positive, and cases have appeared in Perth and Dubbo, New South Wales. The fungus survives in the environment and spreads through contaminated soil rather than primarily through infected animals. Infected lizards have been reported in Brisbane, Perth, and Dubbo, with Brisbane soil samples showing the fungus persists environmentally. The infection begins on the skin and progresses slowly, with environmental exposure and habitat factors playing a major role in transmission over direct contact. NB may exist in the environment without a host and could drive population-wide declines, potentially threatening about 500 of Australia's roughly 1,000 reptile species. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.